So my portion is on RNAi purpose. Now, RNAi is just a term used to describe the cellular mechanism of using double-stranded RNA, chopping that up, and using that to cut off translation of a messenger RNA, and then therefore silencing or knocking down a gene. Now, the reason why scientists want to do this is they want to use RNAi to learn the function of a gene by turning it off. So like, for example, you turn off a certain gene, and the scientists can then figure out what the effect was of that gene on maybe the cellular, tissue, or even organismal level. Now, using siRNA is pretty cheap. It gets easily designed, and it can be administered pretty easily. And there's even some say that it can even be used as a therapeutic tool to fight off infectious diseases or even genetic disorders. And these genetic disorders would have to be due to a result of inappropriate or undesirable genes being turned off or on. Now, RNAi is also used as in normal biological functions, like, for example, defending against transposable elements, maintaining chromosome structure, and regulating developmental timing and differentiation. Now, an analogy would be, like, for example, if you have a cop and he's looking around a parking lot, and he's looking for cars, not a parking lot, a sidewalk, and he's looking for cars that are illegally parked. Now, when he sees a car that's illegally parked, he stops, he puts this thing on the tire that stops it from moving, and therefore you're not allowing the car to drive, which is pretty much its initial purpose. So that cop is essentially just knocking down that car, or the ability for that car to drive.
Hi, I'll be presenting a walkthrough of the method for RNAi knockdown. I broke it up into a few different parts to make it more simple. So for part 1, for RNA inference, I'll be focusing on the in vitro method, which begins with a double-stranded RNA that corresponds to the specific gene sequence of interest. This dsRNA can be formed via in vitro transcription of the sense and antisense copies of the original DNA sequence of the target gene. Below, I've attached a figure from the textbook. Next, the coding sequence of the gene can be derived from either a cDNA clone or from a specific sequence of genomic DNA. One must place this coding sequence next to a promoter and a plasmid vector in 2 different orientations, one normal, one reverse. After transcription of both these constructs via RNA polymerase and ribonucleoside triphosphates, you get RNA copies in the sense and antisense orientations I stated above on the previous slide. Under the appropriate conditions, these 2 RNA copies are able to hybridize together to form the dsRNA. In short, this dsRNA can be cleaved by dicer into siRNA. For the in vitro method in particular, one can inject the gonad of the organism of interest with this newly synthesized dsRNA. Via RNA endonuclease dicer, the dsRNA can be converted to siRNA in the developing embryos. So how does siRNA find the correct corresponding mRNA? It needs help. These newly formed siRNA molecules can cleave mRNA molecules that are of matching sequence. This is done through a reaction that is catalyzed by the protein complex known as RISC. RISC is responsible for helping siRNA to recognize and hybridize its complementary strand of mRNA. Other specific nucleases within the RISC complex aid in cleaving the newly formed siRNA-mRNA hybrid. So the final step and the results of RNAi knockdown are as so. Once the specific siRNA and mRNA sequence is cleaved, the protein that was originally a product of that mRNA is no longer produced. This shows RNA inference has the capability to silence gene functions and is a process that is highly dependent on base pairing specificity. After dsRNA is injected into a specific organism or cell, one should prepare to observe changes in phenotype, function, and or growth. Any change that happens to the cell or organism in the absence of this protein coded by the targeted and destroyed mRNA can give researchers insight on the overall function and role of that protein. There are two ways to examine these changes. The first one is the most common way, which is Western blot analysis, and it's the most commonly used method to detect a change in protein production. Below, I've attached an image that overviews the whole process. The second process is reverse transcription PCR, RT-PCR for short. It's a method that is used to study the mRNA and account for changes in overall protein production. I have also attached a quick figure to overview the entire process.
The research article was looking at red swamp crayfish, which are shown in the picture on the right. They have a gene called the insulin-like androgenic gland gene, known as IAG, which regulates their androgenic gland. This gland regulates sexual differentiation of the male crayfish. IAG can therefore be used for sexual manipulation. The question the researchers had was, how did double-stranded RNA and small interference RNA affect the IAG gene found in the red swamp crayfish? Based on previous studies, they expected that both the double-stranded RNA and small interference RNA should be able to induce RNA knockdown. In order to conduct their research, they gathered adult crayfish from a fish market in Wuhan, China. They were all acclimatized to lab conditions. The researchers then designed 3 different siRNAs and 1 dsRNA, which were synthesized in vitro. 2 different groups of male crayfish were studied, each made up of adults and juveniles. The first group was injected with 3 different siRNAs in 3 different doses, depending on their body weight. The goal of this part was to determine the optimal dosage. The second group was injected with dsRNA with a single dosage, also dependent on body weight. But they were injected once a week for 7 weeks in order to study the long-term effects of RNAi on mRNA levels. The adult crayfish were injected in the cardiocelum, while juveniles were injected in their muscles. After doing this, the crayfish were dissected and samples were taken. RNA was extracted from these samples and used to synthesize cDNA. The cDNA was then diluted and used to run PCR.
This experiment tested the effectiveness of IAG siRNA in gene silencing. After detecting the IAG expression level in the testes, it was found that IAG siRNA3 reduced the transcription level significantly. The 0.5 microgram per gram and the 1 microgram per gram dose greatly inhibited the expression of IAG, whereas the 0.1 microgram per gram dose had a non-significant impact, as can be seen by the figure on the left. siRNA3 inhibited better than the rest, and the 0.5 and 1 microgram per gram inhibited better than the 0.1 microgram per gram. The second part tested the IAG silencing on adult mRNA. After 24 hours, the IAG expression was decreased in the brain, testes, androgenic glands, vas deferens, and the ventral nerve cord. After 72 hours, there was still a significant decrease in gene expression, except in the IAG dsRNA-treated ventral nerve cord. After 120 hours, there was still a significant decrease in expression, except in the siRNA-treated ventral nerve cord and vas deferens. After 10 days, IAG dsRNA continued to inhibit gene expression, whereas siRNA didn't show any decrease in expression. This graph right here explains all the information on the last slide. As you can see, this is 24 hours. siRNA3 and dsRNA are working perfectly fine in silencing gene expression. After 72 hours, dsRNA is not significant. After 120 hours, siRNA is not significant, and neither is the vas deferens. And then you can see at 240 hours that all the siRNA are non-significant right here, and you can see some of the dsRNA, such as the brain, just the brain, are not significant. The third part tested the IAG silencing on juvenile mRNA. Both siRNA and dsRNA were able to inhibit IAG expression in juveniles. dsRNA had significant inhibitory effects on the 7th day, however, siRNA did not. And on the 10th day, both RNA types were ineffective, as can be seen on this graph. The first 24 hours, they were effective, and then in the 72 hours, 120 hours, and at 168, siRNA3 was ineffective. In conclusion, this study concluded that IAG dsRNA silencing lasts much longer than IAG siRNA3. Also, phenotypic changes occurred during prolonged RNAi exposure. Hepatopancreas was damaged in the treated group for males, which eventually caused them to die off. And the treated juveniles expressed appendages for both male and female sex organs, and both of these results caused deaths amongst the tested specimen. Thank you for listening to our presentation.